Good morning, everyone. Am I audible?
Yeah, Akshay, I can hear you.
Okay. So we'll start today's session.
Hello, Kiran.
Hello, good morning.
How are you?
Good. Thank you very much for giving me a time to give talk here.
It's our pleasure. This is the first time we are trying out something where we wanted to just talk about post-image acquisition analysis. And so most of the students have some background in image analysis, at least with PG, I believe. And so we're looking forward to your presentation. And we also like to thank Ganesh for connecting you to us. It's really nice, and I hope on a regular time you would visit Bangalore, India, and we can host you live here. All right. So good luck, and maybe we can get started.
Yes, sure.
Yeah.
Yes, sir.
Introduce.
Good morning, everyone. So we are going to start with the fourth day of the workshop. I am Akshaya, and our first speaker of the day is Akira Saito. Akira Saito graduated from the Tokyo University of Agriculture and Technology. He joined Olympus as an application specialist, biobusiness development, providing support for in vivo imaging system and high content analysis in the year of 2007. He became a product specialist for confocal and multiphoton microscope at Olympus Corporation in the year 2009. He also rendered the role of team leader, supervising sales, promotion, and marketing of global research life science microscopy market. He is currently the assistant manager, life science application and marketing, APAC, Olympus Corporation of Asia Pacific, assisting the head of Olympus Life Science business in countries including India, Australia, New Zealand, South Korea, Southeast Asia, including Taiwan. He is also leading and supporting the product and application specialist in APC for all ranges of microscope, including digital imaging products and software. I will hand over the stage to Tejal so that she can go over the instructions for this session, and then Saito-san, you can start with your presentation.
Yep, thank you very much for the introduction. Good morning, everybody. I am Akira Saito from Olympus Corporation, Asian Pacific. As I was introduced, that I am the some kind of a product, have a product background, and also supporting lots of Olympus microscopes, and also it includes the digital microscopes and also the softwares. So I'm glad to be here today, and I'm very honored to give some talk here. And my presentation will just focus to the 2D segmentation, but not only for the 2D segmentation, but I would like to touch about some kind of new technology which is used for the 2D segmentation by using the artificial intelligence. Actually, at the Olympus has introduced or launched this function in our imaging software CellSense last year, and we have a couple of good feedback from the researchers and market, so that I would like to share some kind of information about it. Okay. By the way, can you see my screen?
Yeah, we can see your
Oh,
screen.
okay. Yeah, great. Thank you very much.
Right.
Okay. So first of all, today's topic is about the 2D imaging, 2D segmentation. But of course, at the for the biological field, we usually use the microscopes, like inverted microscopes over here, or maybe upright microscopes, or confocal imaging system. To have the good image, not only for the good image, but also for the good quantitative information from the biological sample, it needs to have the good optics or good alignment kind of things. Without that, actually, the analysis will not work properly, and the result which you'll get is not will not be as you expected. So it's very important to understand the optics, what kind of objective you are using, and also what kind of microscope technology, I mean the observation methods that you are using. Is very important to understand. And of course, I won't touch about the microscope today. I just only talk about the image analysis today. But I think that the Dr. Ganesh from Olympus, India, has a talk this week, already done this week, about some kind of imaging technology that to have imaging technology talk that what kind of how can I say to get the proper image from the samples. But anyway, today's topic is about the analysis, so that I'll touch about this. And the final goal is that we find out the region of interest. Maybe sometimes I will say like objects that you are focusing on, and and get the information from that object. So actually, there is lots of observation methods in the microscopes. For example, fluorescence in the left hand, bright field by using the some kind of stains and use the color camera, and sometimes that you use the phase contrast or DIC for the to have the contrast. But anyway, the point that you need to understand is that you need to use the observation method, which can detect or make some contrast that you would like to see, or maybe you would like to analyze. So that is the most important thing that you need to think about before going to the image analysis. After the image acquisition, it goes to the image analysis, including some kind of a processing and also segmentation. But anyway, the imaging is actually the most important part to get the quantitative information from the biological sample. So today's topic is about the segmentation of the object. So for example, you got this kind of a nuclear image. This is just the image with the DAPI staining. And you can see that there is a couple of the nuclears are in the image. And the most easiest segmentation is to use the intensity information. I mean that give the threshold that the above some kind of a intensity will be detected. For example, you can just do the intensity thresholding and detect these area. Of course, that if you are not doing proper thresholding, then the detection will be like this, so that it is necessary to have the best thresholding to identify the background, let's say background, and the object. So if you set properly, then it should be like this. However, you can see that some of the nucleus over here is identified as a single object, which you can see like 1, 2, 3, 4, 5, 6, 7 nucleus are there, but it is identified as a one object. To go more how can I say properly segmentation, there is one method called water threshold. Sorry. Sorry. Sorry, I forgot the English. But anyway, there is an algorithm to detect the this peak and gap, and give some how can I say division in the gap. So you can see the peak over here and here and here, and the gap over here and here, so that it will be cut the object over here and here, which is called in our software, we just call like a auto split, but there is this kind of a algorithm in the most of the image analysis softwares. So for the object detection segmentation, first of all, the easiest way is to use the intensity information and do the thresholding with it, and use the this kind of object split method to split the objects that you would like to detect. So of course, after detecting the objects, what you need to do is that you need to have some kind of object analysis. Means that what kind of parameters that you would like to have from that objects. For example, morphology information like area or perimeter, aspect ratio, circularity, such kind of parameters that you can get from the detected objects. And also that you can have some kind of intensity, or sometimes you can say that the brightness of the fluorescence, or maybe color information by using the color cameras. And of course, that you can count the objects, how many objects there are in the image, or sometimes that you can count the, let's say, some kind of spots in the cells or nuclear, such kind of areas. And of course, that you can get the some kind of a position information, like where it is in the image, or maybe well positions of the microplate, distance from the other objects. Such kind of information you can get from the after setting the proper parameters that you would like to analyze from the objects. So and also that if you have any time-lapse imaging, so after the detection of the objects and link that object through the time points, actually that you can do like this kind of tracking and analyze the intensity changes, and also that the how fast it moves or which direction it moves, such kind of thing can be also analyzed by using the tracking. So object detection, segmentation is a very key thing that the for the image analysis, and it will be used to understand or analyze further things from the image. So for sometimes that the for example, this image is mitochondria stained image with fluorescence. You can see that this is the fluorescence image, and use the automatic, not automatic, sorry, intensity thresholding. And you said like you found a good thresholding, but the in this area looks like that the some of the mitochondria is not detected. And if you include this one and put the intensity thresholding a little bit lower to include this this area, then other mitochondria detection will be more bigger, become more bigger, and it might be difficult to identify single mitochondria. So this kind of thing might happen in the image segmentation, and that is if you want to go further, how can I say, more complex segmentation, you can do it, but the you can do it, and you can identify each mitochondria very well. but this kind of issue might happen. And if there is lots of algorithm to identify very complex image, but recently there is a much easier how can I say method for the biologists, because that you can easily see that this is in my we can easily understand that this area has individual mitochondria, and also in the very bright area there is we can identify the single mitochondria very easily, so that the our brain can do it, but if we want to do it in the computers, it is quite challenging things. So that the nowadays that the segmentation is going to use artificial intelligence to identify that the objects what we can understand by our eyes and brain. And also that the in the bright field image, I will say this is DIC image. Sorry, this is the bright field image, not the DIC or phase contrast. But anyway, you can see that there is a couple of cells are over here, but you can also see some dust over here, and the image is uneven. But for us, it's quite easy to identify that where the nucleus are, so that we can use artificial intelligence technology to identify each nucleus, even in this kind of a bright field image. So that this is not the fluorescence image. Sorry, this is the detection result by using the artificial intelligence. But you can see that each nucleus are segmented properly. So for the artificial intelligence segmentation, actually that the deep learning is one of the hot topic in the market for the biological analysis field as well. But for the AI, actually there is some kind of a level for the image detection. One is called deep learning, and this can do this can, how can I say, simulate like our brain, the neurons in our brain, and do some complex detection. And also there is a little bit simpler technology called machine learning, which you can do like just draw the background and also the samples, and then it will detect the sample area in a very short time. So this kind of machine learning is there, but I would like to more dig into the deep learning, because that recently the deep learning technology in the life science microscopy field is increasing a lot. You can see this graph is the number of the publications. I just compare with the light sheet or super resolution, which is the new technology for the observation. But you can clearly see that the deep learning analysis is going more than those kind of a new observation technology, but also that the everybody, all of the biologists are having some issues to detect do the segmentation. And deep learning is doing a good job for the segmentation. So I would just briefly introduce how the deep learning works. So actually for the deep learning, we need to create this kind of a neural network, which is saying that something like our brain, to understand that what the images are, where the objects are. And to train this kind of a neural network, actually we need to have the ground truth and also the image data in very simple way. So this is dog, this is cat, cat, dog, cat. So the image data with the ground truth is required. And bring this one to the neural network training, then the neural network will be trained so that once this kind of another dog image came, then and after doing the analysis of with the deep learning neural network, then it will be estimated that this is dog with the high probability. But sometimes that this tiger image came and use the same deep learning, neural network, then it can be identified as a cat, but maybe it will be a low probability because it is similar to the cat, but not exactly like a cat which is trained by the neural network. And sometimes this kind of a cartoon came in and do the inference by using the neural network, then it will be understand as a cat, but it's similar, but it's different, so it will be as a low probability. So if we include this kind of a cartoon image and give the ground truth and train the neural network, and then of course with the inference with the updated neural network, it will be identified as a cat with the high probability. So the point for the deep learning is that there is a learning phase. I mean that we need to have the ground truth information and also the image information. And after the training of the neural network, it goes to the inference phase to analyze the image, another image, by using the neural network. And also that the inference result means that the result over here will say that this might be cat with the high probability, means that it gives some kind of a probability factor. Let's say in our software, we just say one is the exactly that is the one that the neural network identifies, but it becomes like 0.9, 0.8, 0.7, and if it becomes like 0.3, then hello. Yes.
Excuse me, Saito-san. We have a question in the chat box. Can I take it?
Oh, no? Okay. I thought that maybe you can do the Q&A
if
later.
You are
but
fine, we can take it after the talk also.
Whichever you want. Yes, you can.
Okay, so there is a question. How we will validate the result accuracy? It's from Divya.
Sorry, once again.
I will repeat the question. How we will validate the result accuracy?
Validate the accuracy. So how can I say it? So there is several factors for the neural network itself, whether it is developed very well or not, which I will cover later, by the way. And also that in the inference image, of course, that there, as I mentioned in this slide, it shows that the probability factor, I mean that the 1 or 0.9 kind of thing is there, so that how can I say, we can see how varied it is from the probability factor from the image. I'm not sure whether I am answering correctly or not. Thank you very much. So I will keep continuously doing this my presentation. So segmentation is done in so after the probability map, probability factor map, we can do the segmentation by using that information. I mean that the more than 0.8 is segmented. Such kind of a thresholding can be done. And of course, that the more training data generates more accurate inference, means that the neural network will be more matured. So the last slide was using the very simple cat and dog image, but for our biological samples, I will just use this kind of mitochondria images. So there is a fluorescence image by using the mito tracker, and we need to level that where the mitochondria is, and tell that this is the grand truth of the mitochondria, and combine with the image data over here, and train the neural network. Then the neural network will be created, and then bring to another image and do the inference. Then you can see that the this kind of a result after the inference. This one is the probability map. This means that the red means that it has a high probability, and of course blue is the low one. So that it is so after the inference, it is almost similar way to do the intensity thresholding, which we usually do, so that we need to have some kind of a threshold to segment the object by using the probability map. So for the ground truth labeling, of course, that there's a two-way. Of course, one is that we can use a manual labeling. I mean that identify the background, and I just want to have this kind of a glomeruli information from this kidney section. You just need to mark these kind of a new glomeruli or some kind of object in the image by hand, and create tell that this is the ground truth. And also that for example, if you have a fluorescence image and also bright field image together for the training image, and use the fluorescence image to identify to give the ground truth. I mean that with this ground truth, fluorescence image. So these are the ground truths of the nuclear, and then just apply the inference to the bright field image, then it will be give the probability map, even there is no for DAPI staining, so that it can be the ground truth labeling can be done in both ways. You can use the other observation method, or you can do by your manual labeling. So for example, you have a nuclear over here, a big nuclear over here, and there is some kind of foci in the nuclear, and you can identify the nuclear and the foci, and give the training to the neural network, and identify these two other objects as a different segmentation, even in the single channel fluorescence image. Of course, we can do such kind of thing. And also that we can use the multicolor or multi-channel image to give the more accurate information to the neural network. So that for example, with the single channel image, it was quite difficult to identify this area, but by using the multicolor, like 3-channel image and give the and create the neural network, then it can be identified properly. So I'll cover this one by using our software later. I have a software in my PC so that I can show you how our training will work. But anyway, that we need to have some kind of a training level to identify that what kind of objects they are, and then give to the neural network training. And then after that, even there is no for sense information, we can identify the nucleus, for example. And of course, that it will match with the fluorescence image. So there might be difficult to by using the intensity information or color information to identify the this kind of metaphase of the cells in the phase contrast image. We can use such kind of this kind of a neural network to detect these kind of metaphase, which is going to divide so that we can count very easily even in the phase contrast. I think I got the question now. How far the intensity of the object will training neural network is changing. Sorry. I'll read the question. How far the intensity of the object play role in training neural network? Does change in intensity, suppose low intensity image for same object will have low probability or will be analyzed as accurate as high probability? Actually, it depends. How can I say? Even it is the low contrast, I will say low contrast, not the low intensity, but the low contrast image. And but if you give the ground truth with that image and create the neural network, it will detect even the contrast is low. But of course, that it is better to have the high contrast. But the by using by giving training for the neural network, then it will show a very good result even in the very how can I say the low contrast image. I think I'll cover that one. I have some example later. I think it is in the last part of my slide. But anyway, so for example, if there is a very challenging imaging condition for the bright field, and you can see that there is a high contrast in the center of the well, but the very low contrast in the edge of the wells. But it can be detected properly like this. So there is a very low contrast over here, but it will be detected even with the bright field image by using the neural network, deep learning. And sorry, that the true AI is our name for the deep learning technology. But anyway, when we compare the fluorescence and the nuclear counts by using the fluorescence and also the deep learning technology, just with the bright field, then it is also almost matched to the what we can detect with the fluorescence. So I will say it's quite accurate. So again, sorry, this is the bright field image example to find that where the cells are. And of course, that we can use this kind of analysis segmentation to the time-lapse imaging, and you can find that this is the stem cell, so that the contrast of the background is a bit higher than the regular 2D cell culture. But I can see that it is counted the nucleus very well. So of course that we can use the fluorescence image. Even this is this is level with the mitochondria, but for us it's quite easy to understand where the nuclear is, so that you can very easily understand this is the nuclear, this is one, this. But by using the just the intensity information, it is very difficult to identify where the nuclear is. But of course, by using the neural network segmentation, then we can easily identify that where the nuclear is. Even it is not stained, but we can guess that where the nuclear is. And this one is the one example by using our confocal imaging system. And the customer has 3 different types of the cells. This one is has a intensity signal in the cytoplasm. This one only in the nuclear, but it is almost flat. But in this one, there is some speckle objects inside the nuclear, so that train the neural network to identify these 3 different kind of objects and create the neural network and apply to the real samples that the customers, the researcher want to analyze. Then he can get the very nice result. So this is the training data by using the 100% cytoplasmic AR sample, and this one is the nuclear AR, and this one is the spectral AR, and tell that this one is cytoplasmic, and this one is the nuclear AR, and this is the speckle AR. And by using the validation to those kind of image to see that whether there is a big difference, or find out whether this neural network is valid or not, and also apply to the real sample and see that this one is having more AR speckle images cells compared to this cells, these samples. So this kind of analysis can be done. So we just skip this one. And again, I think this will answer to the question from in the chat. So if we train with the very low, sorry, this right side is the some kind of a very low contrast image, and you can see that these are the nuclear position, but it has a very low contrast, so that of course it is very difficult to identify by using the threshold or other general segmentation. But of course, if we give the ground truth to this condition and do the give the training for the neural network and use it to the other real images, then of course it will be identified very in a good way. But of course, the probability might be a little bit lower than the other high contrast ones. So you can see that this one is very easy to identify. This one is quite difficult, but even in this condition, still that we can segment the nucleus very well. And of course, this is not only for just a single image. You can use it for the very big stitched image as well. And this is just the example to identify the groomery by using this color image. Sorry. And count in this section how many there is and how big it is. It is also possible to use it by using the neural network segmentation. So that is the last slide for my presentation. Before going to the online demonstration, do you have any questions?
Audible? No, Saito-san, I don't think there is any questions in the chat box, but if any
Okay.
participant has any, they can either unmute yourself or just type in the chat box. Saito-san, I think you can go ahead.
Okay. Yep. Thank you very much. Sorry. Okay. Can you see my the software? Or
Yes.
you're saying, oh, okay, good. Okay. So this is the our imaging software called CellSense. And in CellSense, there is a couple of the grade of the software package, software models. And today I would like to use the CellSense Dimension, which is the most advanced imaging software in our analysis part. So maybe you can see that the this image. This is just a simple copy stain nuclear image. As I mentioned, that the easiest thresholding is to use the intensity information, and you can see that it is detected very well. And of course, that you can change the threshold level by yourself by using the by changing the line of this threshold. Okay, and sometimes that there is a automatic threshold kind of thing in the software. Of course, you can use that kind of automatic detection, but of course, sometimes it does not work as you expected. Like for here, you can see that some of the areas that you would like to detect is not detected. So So I'll just come back to the, sorry, there we go. Just come back to the manual thresholding. give some kind of a threshold over here. Sorry. Where this thresholding option is present in cell sense dimension. For cell sense dimension, actually, that there is a two types of the countermeasure thing. You can see that this panel called countermeasure. One is to have the countermeasure basic kind of thing, and another one is optional solution, but which is called countermeasure full option. So anyway, if you have the cell sense dimension, I think you can see this tab, countermeasure tab, and in default there is this kind of a tab in the cell sense dimension. So if you go to this tab, then you can find the countermeasure box in the left bottom side. So this is the default position, but of course that it can be closed. So if you cannot find it, then you need to go to view and find the tool window and go to the countermeasure. And then there is a threshold method by using the automatic, manual, or adaptive threshold. So by using the, if you click manual threshold, then you can have this box, manual threshold, and then you can adjust the threshold intensity, threshold line in this box. Okay, so after doing the threshold, then you can find that the objects are detected, but you can see that there is a couple of the nuclears which are not segmented properly. Let's say there is 1, 2, 3, 4, 5, 6, 7, 8 nuclears over here in one object, so that it is better to split these nuclears, the objects, to have the proper counting for the nuclear. So just select this one and go to the auto split objects. And then you can find that it was okay, say segmented like this, but still that there is one object which is not working splitted well. Then so I'm not sure that why it does not split, but for example, this one, sorry, when you use the auto split, then you can find that all of the nuclears are splitted like this in a proper way. Okay, and you can find that there's other objects which is combined as a one object, so that you can select all of the detected object and use the auto split. And then you can see that it is divided too much. Then I need to go back to this one. Very sorry. Yes, sorry. It is splitted. The nucleus are splitted, and be as a single nucleus like this. So after the segmentation, in our software, you can get the information of the objects like area, perimeters, other green intensity values, maybe shape factors from each object. So for example, okay, object on zoom. Sorry. Plan and zoom. So this one is this one, and this one is this one. This one is this one. So that you can get the some kind of morphology information and also the intensity information. So what kind of parameters you would like to have need to be selected in this window in our software. Of course, that there is lots of parameters that we can get, so that we cannot add all of them. If you add it, then the sheet itself become very big, and it's quite difficult to how can I say to see the one you would like to really focus to, so that you need to find out that what kind of parameters you would like to have from the select object measurement tab in our software. So for example, if you want to add the say irrogation, that which we can see this one, the circle is 1, and if there is a very flat kind of a circle, then it becomes bigger. So it will have a very elonged nuclear or objects. So we just add it over here, and then you can find that information in each object. はい。はい。So, sorry, I got the question. Can we differentiate objects during segmentation as in co-culture? Um, えーっと、So I think if I understand the question properly, the question is that is it possible to identify different kind of cells in the one sample? Okay, so if that is the question, then as an example I showed in my presentation is that so this one, it's not the co-culture, one cell line, but they have a different kind of a structure. And if you give some kind of a ground truth, which cell line is this one, and another cell line is this one, and create a neural network, then it might be possible. But I'm not sure that the how different those two cells are in your co-culture. So I cannot guarantee it will work or not, but I think it's good to try with the neural network segmentation. Okay, so that is the very simple thresholding by using the manual threshold and the parameter setting for the analysis, what kind of quantitative data or value you would like to have from that objects. Another question is that I got is how did you differentiate between class 2 and class 3 cell types have you shown in your image? So I believe what it is over here. So so this is coming from the one of the customers result, and there is an application note in our website, so that I will give you the URL of it later. But what this customer did is that have three different types of the cells that which should have the only have the class 1 cells, and the cells the sample only has a class 2, and this one only has a class 3, and give that where the nucleus are or objects are as a ground truth like this. Sorry that it's quite difficult to see it, but it is labeled the ground truth onto this image and give the training so that so in the neural network it will differentiate that this very flat nuclear or some kind of how can I say spots we can see in the nuclear can be identified in the neural network. I mean, they look, how do you, I mean, they look similar apart from speckling in 3 class image. I am Very sorry, but I cannot understand what you are saying.
Yourself and speaker.
Saito-san, you can probably take up this question later on.
Okay. Oh, Ganesh. Hello. Okay.
Akshaya,
Now,
maybe like I can also help him, Bharti, later on.
Hmm?
So you can put me in touch with her.
okay.
Sure, Ganesh. Sure.
Yeah, thank you very much. So I will just go back to the software. I'll just simply show that how our the neural network deep learning segmentation works. Actually, I cannot show entire process because the training process will give the very long time and also require the very good GPU. So that and I don't have a very good GPU in my hand. It's my laptop here, so that I cannot do it. But I can show that how it works, how you set the training, the ground truth, and give the training to the neural network creation and do the how to do the inference. Okay. So in our software, actually there is this is the optional function, by the way, so that of course that it is not available for all of these thousands dimension. It is need to have the neural network deep learning solution, and if you have that solution, optional solution, then you can find this training level two window. Okay, and okay. どうしようかな。So yeah, I already labeled so that I can I will show you how you can label this one. Okay. Okay, so this is the image with the nuclear and the foci inside the nuclear. And first, I would like to identify where the spots are. I would like to start with that one. You can use the hand level, as I mentioned, by using the I would like to classify that this is the spot. Use this one, select training level. Sorry, no, this one. Create a training level and do the each spots manual handwriting. It is also possible, but I would like to recommend you to use our content measure to identify each spots and then do some kind of a how can I say adjustment by manually. So how to do it is over here, go back to the content measure and adjust the thresholding. And give the content measure. Select. Okay. Then go back to the training. And there is a function called import detected object. Yes. then you can see that these spots are labeled as a number 1. Sorry, I will just change it to spot. And if I think this one is not the one that you would like to have, so that you can just remove it. Oh, sorry. Sorry. Remove it like this, and maybe this one I don't need it. Or if you want to have a little bit more, then you can change the size of the brush over here and put like this. Okay. And I also want to have some kind of a label for the nuclear itself. Actually, that it is a bit difficult to do it by using this threshold, because you can see that it's for this nuclear, I think the nuclear is like oh, sorry, just see. This one is I think that the nuclear is like this, and I think it's quite difficult to use the intensity information to identify that where the nuclear itself is. So I'll just use this and leveling. Okay, so I have two different types of the classes over here. One is for spot and one is for the nuclear. Okay, so after labeling, first it requires to save this image, save as, and oops. So for example, some kind of a truth, save it. And for the training itself, of course, we need to have several tens or several hundreds of images with a ground truth to have the neural network, so that this is I just did for one image, but actually it's takes some time to level for the tens or hundreds images to create the neural network. So once the level is done, then we can go to this one, deep learning UI, and create a new training. Oops. Okay. And you need to select which images you would like to use for the training. For example, I have these 3 images, which is level with the ground truth. So I'll just use this one. And then you can see that there are 2 different types of level in this image file. So I just want to use this one. Would like to train the neural network for these 2 different classes. And there is a couple of types of the neural network. there is different features. Like these two are for the color image, and this one is for the a little bit smaller neural network creation, which can give the result much faster. And actually, our deep learning can be applied to the live image, so that during the live image, we can show that the objects, the segmentation result on the live image, so that if you want to do such kind of live segmentation viewing, then it's necessary to use this fast network algorithm for the neural network. And there is a 3 level of the neural network, which you have some kind of lots of variation of the objects or not. But first of all, you can try with the standard network. And for the training means that the neural network will be created by using the lots of iterations of the training, so that you can try with you can set the duration, how long you would like to do for the neural network training. So I'll just go with no limit. Then it will start to do the neural network training. And I think for me, if I want to do create the neural network with my PC, it will take like a day to do it. So I will stop it. But for the neural network creation, there is one factor called similarity. There is a validation going on to create the neural network to see that whether the neural network is created well or not. And if it becomes close to 1, then it is the neural network is going well, created well. But if there is a very variety of the objects that is labeled, then the similarity cannot be very high. But you can see that with lots of iterations, you can find that the similarity goes to the peak of the peak. Then you can think to stop the training. I'm very sorry that I cannot show this one, but so for example, I will show you how it looks like. So by the way, this is the application note which I showed in my presentation. I think you can find some details in this site. And it's over here. During the neural network creation, you can find this is the similarity, and this is how many iterations we did to create the neural network. And then you can find that it is going up. The similarity is going up, and somewhere over here it is saturated. So maybe you can use the neural network over here to do the iteration. Not iteration, sorry, inference. Just give you the URL of this application note. So anyway, I will stop this training for my for my laptop. Okay, so once it is created, then in our software there will be the neural network file. NN file, which is dedicated for the neural network file format. Then, so for example, this is another similar image. Sorry, same sample with a different position. So, and go to the processing and choose the neural network processing. Then you can have this kind of a box, and need to choose that which neural network you would like to use, and then click OK. Give me 10 more seconds. So it is shown like this. So this is the some kind of a probability map of the foresight. And this is the probability map of the nuclear. So this shows that the different colors to identify the different classes. But you can see that the nuclear is detected properly, and also that you can see that the foresight itself is detected properly. So after this segmentation, then we can go to countermeasure and see that how many to yes. So the thresholding by using the probability map. Just use this one. Change to the ROI and do the manual thresholding to the foresight. Very sorry. So you can find that in this cell ROI 5, there is a 52 foci, and ROI 3, this nuclear has a 121, and you can find that these three nuclears has not many foci as these two. So anyway, the segmentation by using the neural network works like this. And yes, that's all for the online demonstration. If you have any questions, not only for the neural network, any kind of questions regarding our cell sense, I can take it. So if you have any questions, please type in or unmute yourself and ask me.
Saito-san, I have also left my email ID. So in case if any one of them has any questions, they can send me an email.
Thank you very much. Yes, I got the question. Can we also track object using cell sense? Yes, we can. sorry, I need to bring the time-lapse image into my PC, this PC, so that I cannot show it today. But how it works is that first we need to create the objects, and then there is tracking. Tracking is also one of the optional module. But if you have it, then tracking. Yes, here it is. Object tracking. So first it needs to identify the objects, and then filter, if necessary, you need to filter the objects, and then give some kind of what kind of the tracking algorithm you would like to use. I mean, is it moving fast, or is it dividing, or such kind of thing? Need to be set to the algorithm, and then if you click it, click start, then it will give you the result of the tracking. So tracking is, sorry. Track. So this one is also using the cell sense to track the this very low contrast object in somewhere over here, and here is very low, but you can track one object like this. So will we take average of each ROI or itself in final result? And average of each ROI. Okay. So in the our countermeasure result window, we can have the each parameter information over here. And if you want to have the mean intensity of the information, you can find from here. And also that there is in the left you can find the some statistic of all of the ROI information. Okay, so maybe nothing more for the Q&A.
For all the participants, if you have any questions, you can directly unmute yourself and ask. Saito-san, I don't think there are any questions. There are no more questions
Okay.
now.
Yep, thank you very much. So,
Yeah, there is a question.
okay. can we have trial version of the software for hands-on?
Yeah, I can help him. Sajjan Rajput, like you maybe like we can talk, and then in case if you want to do something, maybe like I can be in touch with you.
Actually, there is a 30-day demo license. So that if you want to try it, please contact Ganesh or other Olympus India
Yeah,
members.
Akanksha, what you can do is probably you can take up all these requirement, and in case if they need any help for image analysis, maybe like I can help them.
Sure, sir.
Akshay.
Yeah,
Okay.
no problem.
Thank
You guys can mail to us for any such requests. We will directly convey it to Dr. Ganesh or Saito-san. Saito-san, if you're done with the session, we can take a photograph now.
Photograph.
Yeah.
Photograph.
I mean, the
Okay.
session photograph.
Session photograph.
Yeah, so you can share. All the participants kindly switch on the video. Dr. Ganesh, you too. Thank you so much for the session, Saito-san. We enjoyed it a lot, and I hope all
Thank
the
you very much.
participants also got a hang of it. And yeah, if you guys have any questions, you can either directly mail it to Dr. Ganesh or to us. And yeah.
I think I need. Just a minute.
We are done. Thank
Okay.
you so much.
Yeah, thank you very much. Thank you very much, everybody. Please stay safe.
Thanks a lot. Thanks a lot
Yep.
for your time.
Hmm?
I should thank the Saito-san for accepting our invitation.
Yes.
Yeah, he's
Yeah,
super
no
busy
problem, no problem.
nowadays. He's in charge of Asia Pacific. Like at least we could utilize. He's an expert. We could utilize his time and then expertise.
So Dr. Ganesh is very well, how can I say, high level imaging specialist in India. And he is working with me so that if he has any kind of issue for image analysis or also confocal imaging system, he always come to me so that please contact Ganesh if you have any issues or any questions. I think that Ganesh and I can support you as much as possible.
Thank you. Saito-san is my trainer, my guru.
Thank you, guys. So we will break for lunch now, and the next session will start at 2. So before the session, please download all the files which we have sent in a mail yesterday. Thank you. Bye.
Thank you very much.